Hi, welcome everyone. We're going to run through the steps of meiosis. Meiosis is the second type of cell division, and this is the cell division that is done by sexually reproducing organisms. So these are organisms that need to produce sperm and egg cells in order to reproduce, and meiosis is how they're going to accomplish that. Now, one of the things meiosis produces is genetically different daughter cells. Unlike mitosis, where we go from one parent cell to two daughter cells that are genetically identical, in meiosis, we're going to go from one parent cell to 4 gametes, and those 4 gametes are going to be genetically different from each other. And this is why you can have brothers and sisters that look similar but aren't exactly identical, because the genes that are involved in the cells that are made by meiosis get rearranged, and we're going to talk about that. Now, how does that genetic rearrangement happen? Well, there's a couple things that I'm going to demonstrate for you as I play through this. One of those is crossing over, the other one is independent assortment, and the third is random fertilization. So we're going to see the first two in the video, so let's get started. So what I have drawn out for you right now, I put a little bit of notes in the top corner, hopefully you can see that. But with meiosis, we are cutting the chromosome number in half. So if we have a human cell that would start with 46, by the time that human cell produces an egg cell or a sperm cell, the egg and sperm are only going to have 23 chromosomes each. So we are cutting the number in half. That means that we are going from a diploid cell to a haploid cell. And the haploid cells that we are making are, again, gametes. And again, the important thing with meiosis is that we are rearranging genetic information so that we can increase genetic diversity. All right, so what I have started over here on the left-hand side is a cell that has 4 chromosomes. You can see I have 1, 2, 3, 4 chromosomes. Now, my red and blue chromosome are homologous chromosomes, and my red and green are homologous chromosomes. Homologous chromosomes are chromosomes that are the same size, same shape, and have the same genetic information. And where do we get these? You know, if we were talking about a human cell, humans have a human body cell has 23 pairs of homologous chromosomes. Well, where do those pairs come from? Well, 23 of them came from dad, 23 of them came from mom, to give you a total of 46. So a human diploid number is 46. In the case of the example that we're going to use here, the diploid number is 4. 1, 2, 3, 4, my 4 chromosomes. So I'm going to write up here, diploid number equals 4. We're going to remember that, okay? 2N is the nomenclature for diploid. So 2N, N meaning the total number of different chromosomes, 2N equals 4, I have 4 different chromosomes. Now, before any cell can divide, we have to go through that interphase part, right? We have to go through that DNA replication. So each one of my chromosomes is going to replicate, and that means it's going to make an exact copy of itself. And I'm going to demonstrate that here. So each one of my chromosomes now has replicated itself, and I have an exact duplicate of those chromosomes. That exact duplicate is called a sister chromatid. So right here, sister chromatid, sister chromatid. My yellow, 2 sister chromatids. My green, 2 sister chromatids. My red, 2 sister chromatids. A sister chromatid is an exact copy of the same chromosome. Now, during meiosis has 2 rounds of division. So during meiosis 1, and this step right here is prophase 1, and it's a really important step. In prophase 1, so the chromosomes have already replicated. In prophase 1, these homologous pairs of chromosomes get together in what is called a tetrad. And tetra stands for 4. So a tetrad means a group of 4 chromatids together. Now, an important thing that happens during prophase 1 is that crossing over takes place. Well, what is crossing over? Crossing over is the exchange of genetic information between non-sister chromatids. And this is the first important step to genetic variation during meiosis. So non-sister chromatids get so close together that part of them, they overlap and they exchange some of their genes. Their DNA gets tangled up and they exchange some of their genetic information. I'm going to demonstrate that here. Now, crossing over can take place among all of the chromosome pairs. Okay, you can see I've rearranged some of the genetic information. It can take place along anywhere along the chromosome. It doesn't necessarily have to be just at the bottom or just at the top. It can happen anywhere. But the formation of these tetrads and the crossing over that takes place during prophase 1 is an important, crucial step to creating genetic variation in our daughter cells in the end. All right, so what happens next? So after prophase 1, the next thing that's going to happen is that these homologous pairs are going to line up in the middle of the cell. And I'm going to draw that out now, so I'm going to get rid of some of my things here. They're going to line up in the middle of the cell in what we know as metaphase 1. And in metaphase 1, the homologous pairs line up. So we have our pair here and our second pair, they're lining up. That's metaphase 1. In the next stage of meiosis, we need to split these guys apart. So this is what we call anaphase 1. And in anaphase 1, the pairs are going to split, all right? We would have, I don't have it drawn in here, but in the cell, we would have some spindle fibers that would be attached to these chromosomes. And those spindle fibers are going to separate the pairs. So pairs are separated. Right? So I'll show that again. So this would be metaphase 1. My pairs are lined up in the middle. But during anaphase 1, my pairs separate. Okay, my pairs are separated. So that's anaphase 1. Now, in telophase 1, I'm going to change up my cell a little bit here. Because in telophase 1, what's going to happen is I need to have 2 new cells forming. So once my homologous pairs are pulled apart, I'm going to actually have 2 cells now. So in telophase 1, I've created 2 cells. I went from my homologous pairs being lined up in the middle. In anaphase 1, my homologous pairs split apart. And then in telophase 1 and cytokinesis, I end up with 2 cells. But I'm not finished. I still need to go through meiosis 2. I need to go through a second round of division. So what happens is in meiosis 2, each one of these cells is going to go through the same process at the same time. Between meiosis 1 and meiosis 2, there is no second round of DNA replication because remember, we're trying to cut the DNA in half, so we're not going to replicate it a second time. The only time that DNA is replicated is at the very beginning, right before meiosis starts. So right after telophase 1 and that first round of cytokinesis, the cells are going to enter prophase 2. And in prophase 2, they might just look something similar, just like this, right? The homologous, the pairs are they're just there. And they're going to get ready to move into metaphase 2. And in metaphase 2, they're going to line up again, right along the middle. Okay? So they're lined up right along the middle of the cell. After they're lined up in the middle, we're going to move into anaphase 2, and this is where sister chromatids split. So again, we would have spindle fibers attached to these chromosomes at either end, and those spindle fibers would work to pull the sister chromatids apart from each other. So sister chromatid would split like this, sister chromatid splits, sister chromatid split, sister chromatid splits. Right after anaphase 2, right after those sister chromatids are pulled apart from each other, the cell would move immediately into telophase 2 and a second round of cytokinesis. So if I change my cells up here a little bit, after telophase 2, I'd end up with 1, 2, 3, 4 cells. Notice that in each one of my 4 cells, I have only 2 chromosomes. I started with 4. If you remember, my diploid number was 4. I started with 1 cell that had 4 chromosomes. I concluded with 4 cells that only have 2. So my N number is 2. I cut this chromosome number in half by going through 2 rounds of meiotic divisions. Also notice that all of my daughter cells, which these would be called gametes now, all of my daughter cells have different combinations of genetic material. If you take a close look at the genes or the beads, what is represented as the beads, none of the 4 gametes that were made have the same genetic outcome. They're all different. And this is what would happen in the production of sperm cells, the production of egg cells. All of those cells would have some genetic rearrangement to them, and they would all be genetically unique. So again, 2 rounds of meiosis. We start with a diploid cell that has a full set of chromosomes, and we go to 4 cells or 4 gametes that have half of the chromosome number. We do an important genetic rearrangement during prophase 1 called crossing over, which is important for sexually reproducing organisms. It's what creates genetic variation among the species. Okay, so again, our 4 gametes here are haploid. They have half the genetic information than what we started with. Now, when you have 2 gametes that are both haploid, when they get together at fertilization to make a zygote is a fertilized egg, that is when we get the diploid number restored. So one of the reasons why meiosis has to make gametes that have half the number of chromosomes is because when you have a sex cell and a sperm cell, an egg cell and a sperm cell get together at fertilization, that is where that diploid number is going to come back together, right? So a human sperm cell has 23 chromosomes, a human egg cell has 23 chromosomes, but that fertilized egg is going to have the correct number of 46. If we didn't cut the chromosome number in half, we would have offspring that have increasingly too many chromosomes, and the species would not be survived. All right, so that wraps up a very quick overview of the steps of meiosis. You are going to work through the Explore Learning Gizmo. Working with cats, you're going to diagnose a genetic condition in cats. You're going to study the process of meiosis a little more in depth. But please come back to this video if there's any steps of meiosis that still confuse you.